Hi, welcome. We are gathered here for the December Plant Physiology Focus Issue webinar, and today our topic is root development. We have three speakers and two hosts and a moderator, and I will introduce the moderator who will then take charge of the webinar. So this December focus issue is celebrating root development. It's got some beautiful images in it. It's online now, and the three editors are Sigal Savaldi Goldstein, Krista Testerink, Ji Young Lee, who's here today, and Ikram Blilou, who's also here today. After the webinar, we'll post this to YouTube, and you'll find all the recordings of plant physiology webinars on our YouTube channel. We'd like to thank ASPB members. Their membership dues make this webinar series possible. And if you would like to become a member, you can use the code PRESENTS10 and receive 10% off your membership dues. I'd also like to remind you that the registration and abstract submissions are open for the Plant Biology Meeting 2025, which will be held this year in Milwaukee, Wisconsin. So have a look and consider joining us in Milwaukee next summer. If you have any questions, please type them into the Q&A box, and Hector will be monitoring the questions and asking them after each speaker. Email me if you have any problems, and you might have to dial in again if you have internet connection problems. So now I'm going to turn this over to Hector. Oops, that's the wrong script. Oh, here it is. About to read you my bank bill. Okay, Hector Torres-Martínez is an HHMI postdoctoral associate in Jose Danini's lab at Stanford University. His research explores developmental biology of crown roots in Citeria viridis, aiming to elucidate the fundamental mechanisms governing root system architecture. Dr. Torres-Martínez earned his PhD under the guidance of my old friend, Joseph Dabrowski, where he specialized in lateral root primordium development. So I will turn this over to you, Hector, and thank you very much.
Thank you very much, Mary, for this nice presentation. First of all, I would like to thank the journal Plant Physiology, and of course, Mary, for organizing and coordinating this webinar related to the focus issue on root development. And of course, also I would like to thank the editors, Professor Sigal Savaldi Goldstein from Israel Institute of Technology, Professor Krista Testering from Wageningen University and Research, and Professor Ji-Young Lee from Seoul National University, and finally to Professor Ikram Blilou from Kid Abdullah University of Science and Technology. And of course, for all the effort in preparing this focus issue and make it possible. And moving on with this webinar, our first speaker is Ikram Blilou, who will give us an overview of these focus issues on root development. And first, Ikram received her bachelor's degree from Abdel Malek Essadi University in Morocco and her PhD from the University of Granada in Spain, where she investigated the defensive response of plants to arbuscular mycorrhizal symbiosis. And then she carried out her postdoctoral work in Ben Scherer's lab at the Utrecht University in the Netherlands, where she worked on cell patterning roots. Currently, she's a plant sciences professor at Kid Abdullah University of Science and Technology in Saudi Arabia, and her research focuses on how plant cells communicate to transfer positional information and to instruct specific functions during pattern formation. With that, Ikram, if you are ready to share your screen, please go ahead.
Thank you so much, Hector. And as Mary said, it's a celebration today for root development, and it's something that we really need to cherish because it's not every day that we have a webinar on focus on roots, specialized in root development. So I would like to So I would like first to thank my colleagues, editors, especially Krista, Johnny, and Sigal for including me in this issue. It has been a teamwork. It has been fantastic to team up with them and to collect all the issues and the articles. I also would like to thank Plant Physiology, the team, for their support. They have been great in supporting us at all levels. And a special thanks to the reviewers, because they are the players behind the scenes, and they are the ones who are really making sure that the papers got published, they got in shape, and they bring it to the high standards. And of course, the authors for making the time to write these beautiful reviews and also research articles. So I would like to start by highlighting some of the main articles, and I like the issue, but so please, if you didn't read the article, do so, because they have, it's a really comprehensive collection where we go from macro to micro, going from root architecture to the tissue, response to environment, and to the cell-to-cell communication. So for example, if you go to Wang et al., where they looked at how epigenetic modification based on histone methylation act to regulate patterning, especially in the quiescent center, where we had where we show how cell division in the quiescent center is regulated through histone methylation. Then we have another beautiful article, which is from Imtiaz et al. From Jamie's lab, where they looked at some receptor-like kinases called ERK1, and they show how the division's orientation is controlled by these receptor-like kinases. And of course, Idan is here. He will talk a little bit about mobile signal during root development, focusing on the flash flag model, morphogene gradient, etc., etc. So I will not go much onto it. But then there are other very nice articles where they show how plants are interacting with the environment and how specific tissues are responding to adapt to stress conditions. And in a nice review from Goran and Brady, so what they show very nicely how different tissue types are responsive to either, and how do they respond? For example, epidermis, how they respond, does it respond with hair elongation, allotropism, deposition of cell wall barrier, etc., etc. And we also have, and what I like about this issue is that we kind of went away a little bit from Arabidopsis into more into crops and also to cereals, and then where it shows how environment is impacting development and also tissue patterning. For example, if you look at the response of rice by Liu et al., where they show that the phosphorus availability is limiting the tissue patterning, and it's let's say portrayed by apparition of ironchema cells within the cortical layer. And then we have another complementary article about phosphate starvation and cell stress. We're using transcriptomic data. It seems that there is quite some overlap between responses to stress, to salt stress and phosphate starvation. Then we go to more environment, but also linked to development and root system architecture by looking at how jasmonic acid biosynthesis is linking defense to nutrient availability like ammonium, and this is by Pandey et al. And by Liu et al., so they have looked at how nodules formations affect especially a protein called cystothionein gamma ligase. How does it affect the presence in the nodule root forming nodules are also affecting senescence in shoot wort. And then the other article, which I also liked, which is focusing on some special roots that we don't really hear much about, which are called cluster roots in white lupin, where they show that trialase genes is important for the formation of these cluster roots, and then they really highlight the mechanism behind this formation of these roots. And then we have another nice collection of data that I really strongly advise you to read, which is linking really root patterning with in space. So especially looking at different species, looking at different adaptation strategy, looking at different habitat, going from desert environment to aquatic environment like the mangrove, to also interaction between plant and parasites, and how all this global interaction is shaping the root system architecture. And with this, I really welcome you to this webinar. I hope you will have great time. Please ask questions, be proactive, and enjoy this beautiful collection of talk and these fantastic speakers.
Thank you. Thanks, Ikram, for walking us through these focus issues, and of course, for showing us all these very interesting articles. And with that, I would like to introduce our next speaker, who is Professor Dijun Lee, and she is going to make a dedication to Professor Philip Benfi. After earning her bachelor and master degrees from Seoul National University, Dijun Lee completed her PhD in plant biology at the University of California, Davis, under the mentorship of John Bauman. She then pursued a postdoctoral training with Philip Benfi at Duke University, and nowadays she's a professor at the School of Biological Sciences at Seoul National University, and her research focuses on the regulatory programs that control vascular patterning during root growth, and how these are reprogrammed in response to changing environmental conditions. So, Jijun Lee, if you're ready, please share your screen. Okay, thank you.
Thank you, Héctor. When I joined editor for this focus issue, I never imagined that I would be here today giving dedicated remarks about Philip Benfi, my postdoctoral mentor. So Philip's passing was a tremendous loss to the entire plant biology community, and dedicating this focus issue on root development to him is a way to honor his remarkable contributions to advancing our understanding of root biology and his role in fostering an exceptional scientific community beyond root biology. Studying roots, the hidden half of plant has historically been more challenging than studying above ground structures. However, as shown in this graph, the number of publications on plant roots has surged dramatically in the past 5 years. This upward trend reflects the growing body of knowledge and tools and resources available for root research, and many pioneered by Philip Benfi and his colleagues through their groundbreaking genetic studies of Arabidopsis root development. Their collective efforts have provided answers to several fundamental questions in developmental biology. I don't think I'm listing enough in their contributions, but one of the next major milestones in this field has been the development of a high-resolution root expression data spanning from tissue level to single-cell resolution. So when combined with advances in live imaging, modeling, and other interdisciplinary approaches, these resources are bringing us closer to achieving predictable biology for roots. So I anticipate the upcoming speakers will illuminate some of these exciting developments pioneered by Philip Benfi. So beyond his scientific achievements, Philip was an exceptional mentor, a fact I appreciate even more now than I did before. This graph illustrates the impressive number of Benfi Lab alumni who have gone on to serve in academic institutions, and the diversity among them regarding the geographic origins, race, and gender reflects Philip's commitment to fostering an inclusive and supportive scientific community. So I hope that his legacy as both a scientist and a mentor continues to inspire us to build a more inclusive scientific community while striving for the excellence in research. Thank you very much.
Thank you, Eugene Lee. Of course, also for sharing all this inspiring story about Professor Philip Benfey. Thanks for that. And now continuing with the program, our first speaker to advertise the publication is Andrea Ramirez. Andrea is a PhD student at Stanford University in Josedin in his lab, where she is building a root anatomical atlas of diverse species in the Brassicaceae family to understand how innovation in tissue functions help plants survive under stressful conditions. Previously, she earned her undergraduate degree at UC Santa Cruz in molecular, cell, and developmental biology. However, she was not able to attend today, but fortunately, we have a pre-recorded video where she will walk us through her current research and interest. And I think right now, Mary is going to share her screen. Okay.
Okay, thank you for the introduction, Hector, and hello, everyone. I'm excited to highlight the recent review we wrote for Plant Physiology's focus issue, as well as share some of my own research and future perspectives on root development. So I'll start by showcasing how plants thrive in nearly every type of habitat, from dry landscapes of deserts to flooded or aquatic environments, to the ocean coastlines or tidal rivers of mangrove ecosystems. They can also survive nutrient-poor environments like savannas and tundras, and even off of other plants, like in the case of epiphytes or parasitic plants. And plants don't just exist in these habitats, they actively shape them and contribute to the biodiversity of these delicate ecosystems, which in turn makes these communities more resilient to climatic stressors, including salinity, drought, insects, and pathogens, all of which are predicted to increasingly threaten crop yields in the upcoming years. However, root systems play a central role in how plants perceive their environment, particularly stress signals, and can serve as an avenue for engineering crop improvement. They can rapidly adjust their form and composition to meet environmental demands and perform essential functions such as water and nutrient acquisition. Additionally, roots mediate interactions with soil microbes and other organisms, thus enhancing biodiversity, and they are great carbon sequestrators into the soil and again, ameliorating the impacts of climate change. Given their importance, root systems not only support plant survival, but also serve as valuable models for studying stress responses across diverse species. Despite this, much of our understanding of root growth and function under stress comes from a limited number of model species. Therefore, for our review, we wanted to focus on adaptations in several species across the plant kingdom to reveal variations in anatomy and response to diverse habitats. We also compiled recent studies on the molecular pathways underlying these root adaptations to enhance our understanding of the genes and processes involved in environmental resilience and comment on potential traits for engineering. So I want to share an example from the review of how roots change their anatomy in specific habitats, and in this case, we're looking at nutrient deficient conditions. So this is a depiction of a white lupin plant, which develops cluster roots or proteoid roots under low phosphorates conditions. So cluster roots development is associated with the release of exudates that enhance phosphorus use efficiency and can help recruit beneficial microbes to improve overall nutrient uptake. They do so by developing these rootlets or bottle brush-like outgrowths that arise from the pericycle, similar to lateral roots in Arabidopsis, which you can see here. And rootlets are unique in that they also have determinate meristems, which means that they have a finite growth, and in this case allows for root hairs to form very early on in development, particularly at the root tip here. And this allows for greater surface area for further nutrient uptake. So in this review, we also highlighted comparative studies that reveal possible targets of gene innovation. So for example, studies of, again, white lupin, of the white lupin WOX5 ortholog, WOX5.2-like, which is only expressed in the quiescent center of Arabidopsis, actually shows expanded expression into the vasculature of rootlet meristems in this species. So this makes it a good candidate for future studies to uncover how this expanded expression domain may influence functional innovations like determinate growth in rootlet formation. Of course, there are limitations to these studies. Some of the limitations that arise include the transferability of complex traits across species. We don't always know if the same genetic mechanisms responsible for root changes in one system will necessarily act the same in another system. We also may be looking at gene targets that are involved in other pathways like hormone signaling and thus interrupts downstream and broader functions. But some of the possible solutions include characterizing the evolution of key traits in multiple species to identify better candidates for gene manipulation. Comparative studies on closely related species with different stress tolerances can also ensure greater chances of transferability. So this point brings me to my research. Since throughout my PhD, I've been using the Brassicaceae family as a model to study adaptive stress tolerance because it encompasses a wide range of stress-tolerant species, including halophytes like Eutrema saciginium and Shrinkilla parvula, cold-tolerant species like Arabis alpina, and important bioenergy crops like Brassica napus, Camelina sativa, and pennycress. So all of these species also come from a wide range of environments. And we also have sequenced genomes for these species. Most can be transformed using floral dip, and they are all compatible with high resolution imaging techniques, so their anatomies can be fairly easily visualized. And my observations have led me to focus on a specific root layer called the endodermis, which functions as a barrier for solute and ion uptake. So a little bit more about the developmental trajectory of the root endodermis. So along the root, we have different developmental zones, and the endodermis develops special cell wall modifications, specifically in the differentiation zone. So in the first step of differentiation, we have the development of these Casparian strips, which are lignified structures, and they kind of wrap around the endodermal cells, blocking apoplastic or extracellular traffic. Then towards more mature parts of the root, we start to see the development of the suberin lamellae. So at first, this cell wall modification forms a little bit patchy. So you have a few suberized endodermal cells to begin with. And then when you get to more mature parts of the root, you have a more continuous zone. And this creates this coating around the endodermal cells. So Casparian strips and suberin lamellae are known to act as apoplastic barriers. And out of these two cell wall modifications, previous findings by Marie Barbaron have shown that suberization is a much more dynamic process, and its deposition is dependent on its environment. So under salt treatment, for example, suberin deposition is promoted along the endodermis layer, so you have much higher levels of continuous suberization and smaller regions of patchiness. And this is actually regulated by the stress hormone ABA. Okay, so this is what we see in species like Arabidopsis, which is known to be a salt-sensitive species. But like I said, for my project, I aim to look at multiple Brassicaceae species that have different salt-tolerant, stress-tolerant capabilities. So here I am showing a cross-section of an Arabidopsis root stained with dyes that targets the cellulose and cell walls and suberin. So here are two suberized cells shown, and again, the cell walls in this white color. So when I looked across multiple members of the Brassicaceae family, I started to see variations in suberin patterning. So you can see here that some species seem to have more patchiness versus some species seem to have more continuous superization. However, these cross-sections only give you a snapshot of a specific developmental zone. So I moved on to whole root imaging to better determine variations in suberin patterning. So here I'm showing data of an Arabidopsis seedling stained with suberin, a suberin dye called Nile Red again. And on the left is the root tip, and towards the right is the hypocaudal. So hopefully you can appreciate that Arabidopsis shows higher and earlier induction of suberin levels under salt treatment, which matches what we know again. So what does this look like for the halophytes? So for Eutrema sassaginium, actually under salt treatment, there seems to be not a really big difference in where suberin induction starts. And so these high levels of suberin are maintained. But then when we look at the other halophyte species, Trincula parvula, we actually see a similar response as Arabidopsis. There's this shift in suberin deposition towards the root tip under salt treatment. And this also matches findings from Hongfei Li in this recent publication here. So here I quantified these suberin levels as well into non-suberized zones, patchy zones, and continuous zones. So again, for Arabidopsis, we see that the shift is really apparent here in the amount of continuous suberization. In Eutrema sassaginium, you have a slight shift, but not a very significant shift in suberization under salt treatment. And in Trincula parvula, again, you have a significant shift in the onset of suberization. So like Arabidopsis, it has a greater proportion of the root dedicated to this continuous suberization pattern. So to me, this kind of suggested that these species may have independently evolved mechanisms of salt tolerance. And with parvula showing more plasticity in response to salt, and Eutrema showing high endogenous levels of suberin, demonstrating this kind of pre-acclimated or stress-ready state. So what's next? So to really understand what is going on mechanistically, I will be disrupting suberin biosynthesis across this family with tissue-specific promoters driving a cutinase destruction factor, CDF1, known to deplete suberin and determine what effect this may have on the ability of different species, especially halophytes, to respond to certain stress treatments like salt. So this is just an image of Arabidopsis. Wild type seedlings showing suberin signal, and then these CDF1 lines showing no signal. I have also conducted an RNA-seq experiment to elucidate the genetic pathways that may be involved in the divergent stress responses seen in this family. And it will be interesting to look at also key suberin regulatory genes, especially among halophytes, since they seem to have divergent phenotypic responses. Okay, I am out of time, but with that, thank you so much. I am happy to answer any questions by email, since I'm sorry I couldn't make it in person. Thank you very much.
All right. With that, we will thank to Andrea. Like she said, she couldn't make it to the webinar today, and I already saw a couple of questions in the question and answer box, but we will skip this session for now. But of course, if you are more curious about Andrea's work, or in general Jose's lab's work, please check out her paper for more details. And also, I think you could reach out to her through her email. Okay, our next speaker is Amar Pal Singh, who will talk about the research article on ammonium stress, jasmonate signaling, and root growth. So Amar is a scientist at the National Institute of Plant Genome Research in New Delhi, India. He earned his PhD in biochemistry from Lucknow University at the CSIR National Botanical Research Institute in Lucknow, India. And as a postdoctoral research fellow under Sigal Sebaldi Goldstein at the Israel Institute of Technology, he studied root developmental plasticity in response to nutrient stress. And his current lab is focused on how plants adapt to different nitrogen forms and low nitrogen conditions. By combining physiological, genetics, and biochemical and imaging tools, his team investigates root growth mechanisms and metabolic response on their varying nitrogen availability in plants. So Amar, whenever you're ready, please go ahead.
Okay, so thank you, Hector, for a brief introduction. And before talking, I just would like to thank the editors, reviewers of the Plant Physiology for giving me this opportunity to talk at this platform, and definitely Mary for organizing this webinar series. So today I'm going to discuss about my recent work that is published in Plant Physiology about jasmonate signaling that modulates root growth by suppressing iron accumulation during ammonia stress. So before going into it, just I would like to briefly introduce what we are doing inside the lab here. So the overall aim of our lab is to improve the nitrogen uptake and use efficiency by modulating root system architecture, and with a special emphasis on interplay of growth hormones. So we are using three different systems: Arabidopsis to understand the basic mechanism, how do plants adapt to nitrogen deprivation or different nitrogen forms. And this is the tomato work, where we have shown that this enhanced BR signaling leads to improved root foraging response, which is associated with improved plant growth as well as yield. And we are also working with the rice, where we wanted to decipher how this root system architecture changes in response to various nutrient stress conditions. So now we know that nitrogen, it is a complex nutrient and essential for plant growth. So today, mostly I will be discussing about the genetic basis, how this ammonium stress regulates root growth development. So nitrogen is one of the most abundant molecules that is present in the atmosphere, but plants are unable to take it. So the major nitrogen, inorganic nitrogen forms that are nitrate or ammonium, they are taken up by the plants with high and low affinity nitrate and ammonium transporters. So we can see that this ammonium is converted to nitrite and then ultimately nitrate, and plants are able to uptake it via nitrate transporters. However, plants can directly take it ammonium from ammonium transporters. But the major problem is that these the nitrate is like less reactive and it is leaching into the groundwater. So our almost 50 to 60% nitrogen is lost when we are applying to the field condition. So we can see here that these two nitrogen forms, ammonium and nitrate, they have contrasting effect on root system architecture, where nitrate has longer primary root as ammonium suppresses this root. Despite the fact that this nitrogen forms, the availability of nitrogen from low to mild or deficiency has dramatic effect on root system architecture. We can see change in lateral root length, elongation, as well as number. So this is called the foraging response of the plant that adopt under adverse nitrogen environment. So if we talk about the ammonium, so ammonium is the fertilizers with high ammonium content leads to the soil acidity, and this causes leads to the ammonium toxicity. So if there is like soil acidity or there is like acidification of the soil, then the plants face different challenges, that is availability of different minerals for the plant. So we can see in acidic soil there is like high content, the iron is more prone for plant uptake. So this leads to the basis of this work, that whether these ammonium and iron are connected, and we can see here ammonium leads to the suppression of root growth, whereas nitrate is promoting it. So now we want to understand how this ammonium and iron is modulating this root growth. So it is well established that low iron is able to promote primary root elongation under ammonium stress. And indeed, in our experimental setup, we found that low iron is able to promote primary root elongation in Arabidopsis. And by using microscopy, we have found that both meristematic cell divisions as well as cell elongation is affected in ammonium-grown roots, whereas iron is able to enhance it. So this correlates with the enhanced primary root elongation in during ammonium stress. And this is, again, we wanted to understand how this is happening. So it is really due to the high iron accumulation. So we can see in ammonium stress, there is high iron accumulation in the root. However, low iron is able to suppress it. So, and this is correlated with the increased ammonium content under the similar condition. So now if there is high ammonium, then it will lead to the increased iron content. So we wanted to understand how this iron availability regulates the genes involved in iron and iron response during ammonium stress. So we performed the RNA-seq experiment, and we found that several genes are differentially regulated by iron availability under ammonium stress, and some of the key genes which are indeed involved in iron uptake from the rhizosphere, they are also altered by ammonium availability. So this suggested that yes, indeed, iron is playing an important role during ammonium stress. So when we did the pathway enrichment analysis, we found that one of the hormone, that is jasmonic acid, it is coming at the top when we compare the DEGs between ammonium versus ammonium minus iron. And we can see the several genes that are well demonstrated to regulate the jasmonic acid response. They are differentially regulated by iron availability. So this led us to make a background that most probably this is the JA which is governing the ammonium stress response of the plant. So first we tested the hypothesis whether if this is JA dependent, whether JA can promote the primary root elongation during ammonium stress. So indeed, when we applied exogenous JA, we can see that with respect to ammonium, JA was able to promote primary root elongation. And further JA quantification also suggested that low iron is able to increase the JA content in ammonium grown roots. So far what we found that that ammonium is suppressing JA content, however, low iron is able to suppress ammonium content. So now, if this is indeed associated with the cell division rates as well as cell elongation rate, so we applied JA from outside, and we can see in case of ammonium, when there is a small meristem, JA was able to enhance the meristematic zone region in ammonium grown seedlings. And this is again associated with enhanced cell division activity by using this cyclin DB marker genes. So this clearly suggested that, okay, indeed JA is able to maintain the meristematic zone activity, and further JA is again able to suppress the iron content even in the presence of ammonium. So this data, these observations clearly suggested us that, okay, this is the JA that could be potentially regulating this ammonium and iron homeostasis in the plants to govern the root growth. So if we see this JA biosynthesis and signaling pathway, we can see that one of the major transcription factor, that is MYC2, and this is governing several growth and defense responses in the plant. So we looked our RNA-seq data set, and we found that in low iron condition, this MYC2 is enhanced even under ammonium stress conditions. So this suggested that maybe this is MYC2 dependent response. So for this, we use the MYC2 loss of function mutant, and we can see the loss of function mutant root length was less responsive to low iron mediated PR elongation, and this is also associated with the reduced meristem activity in case of low iron condition as compared to the wild type. So this data led us to understand that, okay, this is the MYC2 that is governing this root growth recovery under low iron condition. So now the question comes, how this MYC2 is governing this root growth. So is it regulating the iron content and ammonium content? So indeed, in MYC2 mutant, we can see a higher level of iron accumulation. And not only in MYC2, the loss of function mutant of COI1, that is JA receptor, it also accumulated a higher iron content. And indeed, this iron accumulation is associated with the higher ammonium content under some similar conditions. So from here we can say that okay, ammonium leads to the acidification of the rhizosphere, that leads to the increased iron availability to the plant, that in turn suppresses JA biosynthesis, and once JA biosynthesis is suppressed, this MIC2 is suppressed. So now the question comes, how this MIC2 is governing this ammonium and iron balance. So we searched our RNA sequencing data set, and we found that the one of the nitrate transporter, that is NRT 1.1, it is enhanced under ammonium stress condition, although the plants are suffering due to high ammonium levels. So this led us to investigate whether this is the downstream candidate of MIC2 that is governing this ammonium and iron response via JA. So first we evaluated the expression of this NRT 1.1 via JA and as well as in MIC2. So we can see in MIC2 mutant, this NRT 1.1 is enhanced even in both ammonium and ammonium minus iron condition as compared to the wild type plant. So this clearly suggested that JA is somehow involved in regulating this NRT 1.1. So then to evaluate, we first analyzed the promoter sequence of this NRT 1.1, and we found that there are putative MIC2 binding sites in the promoter of this gene, and we validated the binding of this MIC2 with NRT 1.1 promoter, and indeed in binding assays by using GPRMSA, this MIC2 is able to bind with the NRT 1.1 promoter, and in repression assay it is suppressing the NRT 1.1 function. So now what we understand that this low iron in suppresses ammonium content, and this ammonium leads to the inhibition of JA level, and now MIC2 can directly regulate the NRT 1.1 by binding to its promoter. So what happens to the balance, what happens to the iron and ammonium homeostasis in these plants? So first we raised the overexpression lines of NRT 1.1, and we tested the iron and nitrogen responsive genes, whether they are altered in this in high NRT 1.1 line. So we can see that most of the genes that are involved in iron response or nitrogen response, they are misregulated in overexpression line. And we can see here in NRT 1.1, when they reach like both nitrate and ammonium condition, the nitrogen responsive genes are quite high. So this suggested that, okay, NRT 1.1 is involved in modulating the gene expression. So what happens, whether it is also involved in enhancing the iron and ammonium content. So we can see this NRT 1.1 leads to enhanced ammonium content, iron content, as well as ammonium content in under ammonium stress conditions. So this suggested that, okay, NRT 1.1 is one of the player that is governing this ammonium and iron status of the plant. So now the question comes whether enhanced ammonium and Fe iron in NRT 1.1 alters J signaling and response. So it could be a feedback mechanism. So how this is happening? So for this, first we tested the JA response gene, and we can see the overexpression of NRT 1.1 leads to the partial suppression of the JA biosynthesis genes, and the negative regulators are differentially regulated. And if this is the case, then what happens to the JA levels in this plant? So we can see both JA and JA isolation, they are reduced in overexpression line. However, the low iron was partially able to recover them. So now the question comes whether this enhanced iron and ammonium accumulation via NRT 1.1 leading to a feedback regulation, thus suppressing the JA response, which is associated with primary root growth under ammonium stress. So if this is the case, then how this JA is regulating this ammonium and iron status in overexpression line? So we can see here, as we saw previously, that when we are putting JA in ammonium grown roots, it suppresses the JA content, iron content. However, the overexpression line shows compromised response, and the suppression is not as striking as we can see in case of wild type. So this suggested that the part of response is governed by additional factors, and it's not dependent on NRT 1.1. And here similarly we can see in quantification data where ammonium under ammonium condition, the NRT 1.1 shows a higher iron accumulation, and even in JA condition it shows reduced, but it's not comparable to the wild type. So it's like feedback regulation, that when there is high ammonium and iron, then it will lead to activation of this suppression of JA content, and in turn it is leading to the accumulation of more iron and ammonium and suppressing the root growth. So since this model based on that it is ammonium dependent iron accumulation that is modulating this root growth. So whether this is specific to ammonium or it is specific to other nitrogen sources. So we evaluated the phenotype when we used both nitrate and ammonium, and we can see the roots are smaller. But if we reduce it, then again we can see the roots are longer. So this suggested that this response is associated with high nitrogen content. And then further we evaluated this phenotype by using specific nitrogen source, that is nitrate, and we don't see any surprising difference in the root growth inhibition in nitrate condition. Only the difference can be seen in case of ammonium. So this is like ammonium specific phenotype that we see in overexpression of NRT 1.1 line. So now the thing is that this high ammonium leads to the rhizosphere acidification. So if this is the rhizosphere acidification, then if we increase the pH of the medium, the phenotype should be reversed. So we found that indeed when we are increasing the pH compared to the acidic pH, we don't see a much difference in the root growth inhibition in overexpression line also. So this leads to hypothesize the model that final model that when there is ammonium will enhance the rhizosphere acidification that causes high iron accumulation, that in turn inhibits JA biosynthesis and signaling. And this MYC2 transcription factor, that is the positive regulator of JA, suppresses NRT 1.1. And if NRT 1.1 active, it will lead to the accumulation of ammonium and iron that ultimately we need with the JA biosynthesis and signaling. So in this way, this loop is governing the primary root elongation during ammonium stress. So now the open question is whether JA response is associated with FE translocation in plants, so that we are working and we are trying to understand how JA is regulating the FE status of the plant. And the second, how does N as a sole ammonium source enhances FE uptake and homeostasis in crop plant? This we need to understand how it is happening, and then how this JA governs ammonium response in crop plants. So these are the open questions that we are trying to understand in the laboratory. So with this, I would like to thank my group, and this work is being performed by Anshika Pandey and our collaborators and funding agency. And thank you, Plant Physiology, for giving me this opportunity to speak at this beautiful platform. And thank you, editor and Mary.
Thank you so much, Amar. Very exciting results. Let me check the question and answer box here. I think we have a couple of questions. Yes. First one is from Majid Ali. How do soil nitrogen levels and ammonium concentrations impact root hair development and function? You have
yes,
taken a look at it.
Yes, so the root hair development is definitely impacted by this rhizosphere pH. That leads to the, like if there is like acidic pH, it is changing the root hair spatiotemporal distribution of the root hairs. Because if there is more uptake of the ammonium, the plant is dying. So somehow it is like suppressing and the development of the elongation of the root hairs. So, but so far there is not I don't see that there is a solid evidence that how this is happening in terms of root hair. There is a lot of evidence that lateral roots, yes, the density is increasing because of the lateral roots are smaller. But for root hairs, there is not a solid evidence so far, in my knowledge.
Mm-hmm. Mm-hmm. Nice. Actually, yeah, the second one has to do with lateral roots. So how do jasmonate mediate changes in root system architecture, specifically alterations in lateral root density and primary root elongation, contribute to ammonium stress tolerance in Arabidopsis thaliana?
Yes, I agree. So we focused on the primary root, but we haven't looked here at the lateral root. But someone has to look, definitely. But we focused on primary root, and we can answer that yes, jasmonate is governing this primary root by modulating cell division as well as elongation. So in terms of primary root, yes, but lateral root, we haven't yet checked. Thank you for suggestion.
Nice. Okay, with that, thank you so much, Amar. I guess if there are more questions, you could reach out later to Amar via email. Thanks, Amar. For now, we should move on to our next speaker. Our next speaker is Idan Efroni, who is presenting an update about morphogenetic signals and root patterning. And Idan is an associate professor at the Institute of Plant Sciences and Genetics at the Hebrew University of Jerusalem, Israel. Idan completed his PhD with Yuval Shed at the Weizmann Institute, focusing on factors regulating leaf growth, and then he conducted his postdoctoral research in Kenneth Birnbaum's lab at the New York University, where they applied single-cell technologies to study plant regeneration. In 2016, he established his lab at the Hebrew University, and in 2017, he was recognized as an international research scholar by the Howard Hughes Medical Institute. Idan's research investigates the mechanisms guiding plant organ formation, repair, and the evolutionary adaptations to these traits. And he also explores the ancient cis regulatory code that drives phenotypic diversity in angiosperms with a focus on its molecular evolution and developmental significance. And with that, we pass the microphone over Idan. Please, then, whenever you're ready.
Okay, so thank you very much. I'm going to talk about the most important question in the world, is how do you know who you are? And that is the most important question in the world, whether you are a cell in a tissue or whether you're a human being in this world. And you know, it's the most important question in the world that if you go to ancient Greece at Delphi, the temple of Apollo, it had maxims inscribed on the walls. And the first one and the most important one was know thyself, know who you are. So how do you know who you are, right? That's a good question. And again, it's a question for the cells and it's a question for humans. But for the cells, the way that they know who they are is they have basically two things that they know. They know where they are and they know where they came from. And together that makes who they are, right? Where they are in the tissues and where they came from. So what cells originate them. You can argue that's also true for humans in large extent. But then this leads to the question of, you know where you are if you know where you are, right? And where you came from. And we know that in plants, at least, these two information points don't carry the same weight. So knowing where you are is much more important for your identity than what is your progenitor cell, where you came from in terms of where you divided. So basically, if you look at the plant tissues, the most important question to answer for who you are, what identity to become, is where you are. So now the question is, how do you know where you are, right? You're a cell in a tissue. You're surrounded by cells, and the way to know where you are and the way that plants do it is by asking around, right? Looking around at your surrounding and asking what are the signals that are around me? What do my neighbors basically tell me about where I am? So the way that this is molecularly accomplished is by a range of molecules, which broadly we call the morphogens, that molecules transmit between that cell transmit between each others in order to inform them where they are within the tissue and then to tell them what identity to establish. And the best model case for that in plants came from Phil Benfey's lab, and that is the best described case, and that is the case of short root. Again, this work was pioneered by Phil Benfey, and who's my scientific grandfather. And he's watching here over all the research, so I hope I don't make my mistake. But basically, the short root model works like this. We have a transcription factor that's encoded within the steel, within these eternal tissues. It moves one layer and then stops, and then defines all these cells around it, the quiescent center, the endodermis, gives them the information that they know now they're surrounding the steel, and therefore they should adopt the different tissues. And there are many other mobile signals that together integrate to tell these cells where they are within these tissues. So just one example, because we're finding out these signals every day. Here's an example of not a transcription factor, but the microRNA. So this is a microRNA 160. Again, it's transcribed in the steel. But if you look at its reporter, so this is basically a GFP with the microRNA recognition site, you see that the effect is much broader, and the microRNA actually moves from where it's transcribed to one cell file to the QC, where it can regulate other functions. Okay, so these signals move, but one of the important things about the movement of these signals, that you have to know when to stop, right? You want to translate the signal from one cell to another, but the signal has to stop somewhere, or else it will keep on going on and on and on, and you're not going to get the information that you need. And that leads to, by the way, to the second maxim of the Apollo temple in Delphi, that says nothing in excess, nothing too much. So that shows you how important these things are. And the way at least it works in the short root is that there is a trapping mechanism. So short root moves one cell and then gets trapped in the cells in which it moves to. And the original hypothesis was that it's trapped by binding another transcription factor, scarp root, which limits it to the nucleus and prevent further movement. But we know now that this is much more complex. There are other mechanisms at play, and this is at least gives some of the pictures, some of the answer, but not all of them. And what I'm going to talk about now is a little bit of a work that we've been doing. We've been trying to ask these questions of how do these mobile signals move within, and how they are defined within the root, and how their mobility is defined within the root, and how do cells use this information in order to change control their identity. And the system that we've worked on to try and understand that is that of the root regeneration. And this is a very curious system. You can cut the tip of the root and the root will reform. And the reason that we're interested in the case of mobile signals is because the root will reform from cells right here at the tip. So you have all these mobile signals telling cells what to be and where they are. But once we cut this, these cells have to somehow reset all these mobile signals, and they have to adopt a new identity in order to form the new root cap and repair the root. So we wanted to know what goes on in this specific region in terms of the mobile signals. And we used quite a well-established system where we let GFP produced above the meristem here, and then moves freely throughout the meristem. So you could see basically the movement patterns of non-targeted effects. So things are just passively moving through the meristem. And what was surprising was when we looked at this specific region right after the cut, so between 16 and 24 hours, we saw that in this region, there is an inhibition of movement, and this is just passive movement. So things are not coming into this region. So this suggests that maybe during regeneration, you want to somehow block these mobile signals or block something coming out in order to reestablish your identity. So in order to find out whether this is actually required and what regulates this, we isolated these cells right at the tip using single cell, and we looked for genes that are upregulated specifically in this region in response to the cut. And then we overexpressed many in a screen, we overexpressed these factors and looked what factors can stop the movement. And okay, we looked at a lot. I'll just show you a group of genes that we found. It's a group of DNA binding lateral organ boundary genes. And when you overexpress these genes, they are able to stop movement. They stop the passive movement, but they also stop active movement, which I'm showing you here. This is short root, and then we induce the gene, and we start to see these holes in the movement of short root, which later leads to division, which are characteristic for a low level of short root. So these genes can stop the movement. What happens if we knock them out? So it's a group of genes of five actually LBDs that are co-expressed. If you knock out individual ones or double ones, there's not much effect. But once you remove 4 of these genes, we can see that at least the distal part of the root is disrupted in terms of its organization. But what's more important now, when we make the cut and we look at what's happening to these region with our free-flowing GFP, we see that GFP can now enter this restricted zone, right? So GFP can now enter the zone, so we don't have this block on the movement at the tip. And this is true also for other factors that we've looked at. So this is, for example, short root. Short root, again, transcribed here. But then if you look at the protein does not enter this zone. It can move to the side, but it doesn't enter at the bottom. But in the mutant, it just goes all the way down, doesn't enter the nucleus, but it's able to penetrate this regeneration zone. Also, we looked here just for completion, the microRNA, the miR-160, that's here, it's restricted, like you saw before, and here in the mutant, it's spreading all the way basic to the tip. So what happens to regeneration in these mutants? What happens is that it starts fine. It starts perfectly fine. They start to divide, but then at a certain point, and that coincides with the time where we see the activation of these LBDs, that we start to see these abnormal cell division. So cell division go array, and instead of recovering these identities, these cells kind of divide pretty much randomly, and we get this block and failure to regenerate. So what we think is happening, and this is just a little bit of a taste. The paper is on bioRxiv if some of you are interested. And we know that if we restore the block to this region, so if we activate PDLP, the protein that blocks the movement of signals into the regeneration zone, we can recover the regeneration. So this is an important part, at least in the regeneration. But the question now, what do these protein also, do they act only during regeneration? And the answer is we don't think so. So normally these group of LBDs, so LBDs are pretty widely expressed, but these specific LBDs are located at the root cap and epidermis. And normally, if you follow the division, I've shown you that in the mutant, we see this disruption in the distal part. And when we look at the movement of multiple mobile signals, and this is again just showing short root, we see that what happens is that these signals don't stop, right? So this is short root, again, in the wild type, moves to the QC and stop. This is in the mutant, even though the promoter stays the same, it's unaffected. And we tested that. We see that to the epidermis, everything is moving fine. But then if you look at the cap, you start to see short root cytoplasmic signals basically permeating the cap itself. And again, this is true. We looked at multiple other signals. They're all able to move through the QC and then through the cap. And what we think is that this inability to mark this location, to block the movement, is what's leading to the disruption in patterning, both in wild type and regeneration. So this tells you it's important when to stop. And here I'll abide my own advice, and I'll stop here. Just thank the people in the lab, and this was mainly the lab of one PhD students, Itai Cohen, and I'll be happy to take any question if there are any.
Yes, thank you so much, Ellen. Really, really nice presentation. Let me see the question box.
Can I ask a question, Héctor?
Sure, Mary?
Ikram. Mary?
Ikram, of course. Sure, go ahead, Ikram.
Beautiful talk, Idan. It's really, really nice and really exciting to see shortwood moving downward and not laterally, right? It's really beautiful. So have you looked at cyclin D6 and what are the identity of the cells when triggered by these new divided cells from shortwood?
So we haven't looked at cyclin D6, but what we see is where you see short root, you lose the cap markers. So you lose columella and cap markers, but you don't get activation of steel markers. But you don't see nuclear short root in these cells. So you see cytoplasmic short root. So I don't know if short root is actually able to activate its targets. At least when we look at transcriptome, we don't see ectopic activation of short root targets in these cells. So we think you need something else, right? The ability probably to activate scarecrow in order to be able to activate target cells.
No, it's really beautiful. And the shape of the cells are, well, maybe you can talk about it later. Sorry, let's give the room to others to ask questions. Thank you so much.
Okay, here I have another question from Jasmine Reyes. She says, are these LBD genes specifics then from root? Do you think they could be also controlling, for instance, movement from leaves towards roots or in other contexts?
So these are not specific to roots. So we isolated this specific 5 because they're induced during regeneration, but many are broadly expressed. So some of them are expressed in the shoot tissues, and we see some shoot phenotypes. But we don't think these are important for long-range transport, but rather short-range, so just cell-to-cell communication. And I haven't in time to show, but if you induce these genes, that would lead to callose accumulation at plasmodesmata. So we think that's probably how they act. And if you look at the mutant, there's reduced callose accumulation. So it's probably cell-to-cell communication.
Thank you so much. I think there are no more questions here. So probably actually we are running out of time now. So with that, I would say thank you so much. Thank you so much for to the speakers for their time too. And yeah, I don't know, Mary.
Thank you, editors. Thank you, speakers. Fantastic talks. Thank you, participants. And thank you, Hector. You did a great job. And we'll see you all in January when we're talking about hypoxia in plants, which should be another fun one. Thank you again. Bye-bye, everybody.
Thank you.